Okay, so why don't we go ahead and get started? Welcome to the International Webinar Series of the University of Maryland-based Global Future Alliance. We are delighted that we have so many folks joining us today, and I'm so happy to introduce Dr. Heather Amato, who will be our speaker. Dr. Amato is an environmental epidemiologist who investigates microbial pathogen transmission through a One Health perspective. She employs spatial and molecular epidemiology to assess interventions like water, sanitation, hygiene, and mass drug administration aimed at reducing pathogen spread and antimicrobial resistance. Her research integrates environmental pathogen surveillance and community knowledge to identify key exposure pathways among vulnerable populations, including mothers, young children, people experiencing homelessness, and farm workers. Dr. Amato collaborates with partners in Nepal, Benin, India, and the US, addressing antimicrobial resistance and other pressing global health challenges to improve outcomes for at-risk communities worldwide. So please join me in welcoming Dr. Amato.
Well, thank you so much for having me this morning. I really appreciate it, and look forward to hearing some thoughts and feedback and having some discussion with everyone here about this work. And thank you for the introduction, Dr. Subkota. So today I'll be talking about research that's ongoing in subsistence farming regions in Nepal outside of Kathmandu. And the focus of this work is really on animal waste management and antibiotic-resistant bacteria in these households. And this project actually originated when I was previously at UC Berkeley, actually a long time ago when I was a doctoral student, and then moving into a postdoc, and even now. So this work has really grown over the last several years, as has this partnership with our collaborators at Dulakil Hospital in Nepal. So we still have lots of partners at different institutions, as everyone has since left Berkeley and gone on to doing other great things. So as I mentioned, this project is really focused on animal waste management, and this is in the top right. This is a map of the study area. So Kathmandu is the purple dot in Nepal. We're really working in this Kaveri Palanchok district in the sort of pinkish red area right there. And this is a subsistence farming area. And so a lot of the households have livestock, poultry, and other domestic animals. And so there are a lot of different ways that people handle and manage their animal waste. And they're dependent on the species type, as we'll get into the animal species type, but we have composting piles, we have anaerobic biodigesters, there are different confinement methods like animal sheds, and then a lot of the animal waste is used to apply to fertilizer or to apply to crops as fertilizer. So this is not only are the animals helpful in terms of food security and providing a bit extra income, but they're also providing a valuable resource in the fertilizer for their household crops. I'm going to specifically talk about these anaerobic biodigesters because I think this is a really interesting component and interesting contextual aspect of this study area. And so specifically these biodigesters, I'll get into the details a little bit here. The goal of these household biodigester systems is to replace wood-burning stoves to reduce household air pollution and the risk of respiratory infections. And the way that they work is they basically contain both human waste that it's piped directly from latrines into these underground pits. And then we can also add animal waste into these inlets here. And in these underground tanks, the manure and feces is composted, and it produces methane at the top of this tank that is then piped into the household for clean cooking fuel. The composted manure can also be used as fertilizer then. The effluent that comes out from the effluent pipe here can then be placed onto crops as a really rich fertilizer. So this is a really holistic intervention, and Nepal and other countries globally have really, over the last few decades, expanded these biodigesters across rural communities that have issues with energy access and sanitation and hygiene. And so it's viewed as a really holistic intervention, and it has myriad benefits. So it can also help reduce deforestation. It captures and reuses methane, as I mentioned, reduces costs for firewood, and theoretically, it should be containing fecal waste that would otherwise be left in the environment through open defecation or from the animals, potentially. Um, if that animal waste is not contained and just left out in the environment. So ideally, this holistic intervention should also be containing fecal waste and preventing exposures to harmful fecal pathogens. However, we initially found just through observations, through actually other research that was really focused on respiratory infections and household air pollution. We just kind of anecdotally observed folks interacting with the animal waste for managing these biodigester systems. And we found that there might actually be potential sort of side effects of these interventions that are supposed to be public health interventions. So there might be these unintended consequences of using these biodigester systems. And in a secondary analysis from a repeated measure study of over 500 households, we identified that using these biodigester systems actually increased child diarrhea risk by 31%. And so because this was a secondary analysis, we didn't really have a lot of understanding of the mechanisms and the potential direct exposure pathways. We just knew a household used these biodigesters and then they had an increased risk of diarrhea, but we didn't really understand what was going on. We hypothesized that maybe there were increased fecal pathogen exposures through caregiver hands. Female caregivers were primarily responsible for managing the animal waste and using these biodigesters. And we observed through some qualitative work, interviews and video observations, we observed some caregivers would be working with the animal waste and then directly go maybe just rinse their hands with water quickly or not at all, and then go handle food or handle their young child directly afterwards. So there might just be some contact and exposure through caregiver hands. Mostly because of collecting animal feces by hand to put into these inlets, shown in the pictures here, where then the waste is mixed with water and urine, and then it goes down into the underground pit. And so we were hypothesizing that collecting and mixing the animal waste by hand was potentially a cause for concern and a source of contamination. In the pictures on the bottom left here, this shows on the far left, we see an inlet tank where animal waste should go. This was right after construction. And there are these metal agitators inside the tank that should have a handle on top, and you should be able to just rotate the handle, and it should scrape any solids off the bottom and really help all of that waste go down into the tank through that hole at the bottom. And unfortunately, there were a lot of missing parts, missing or broken pieces that prevented the caregivers from being able to actually use these mechanisms that are supposed to prevent them from having to use their hands. So I am going to show a quick video of what this actually ends up looking like. So we see this caregiver is actively mixing slurry and she has to remove the solids with her hand. She really has to get in there, and hopefully everyone's already done eating breakfast. I apologize. This is always um, a fun video. But the point is really that there is a lot of direct contact with this waste, despite the fact that these interventions built maybe 20 years ago had this in mind where, oh, we're going to build this device in the middle that has a handle so that they don't have to do this, and they'll just turn the handle and it'll scrape everything out. But things break. There's lack of maintenance and continued investment in these communities. So this is what we're seeing now. Um, so I just wanted to touch on why exposures to animal waste also indicate exposures to antimicrobial resistance in this context. We know that there's a lot of antibiotic use in the Kathmandu Valley, including the use of last-line drugs like colistin. These are commonly used in poultry production in the region. And there's also pretty low knowledge and practices around antibiotic stewardship in the region, as reported in previous work. So we also know that antibiotic resistance or antimicrobial resistance in Nepal is an issue, as it is globally. AMR has been detected in humans, livestock, and poultry in urban areas. And there's also a high prevalence of multi-drug resistant infections, specifically extended spectrum beta lactamase or ESBL. So extended spectrum beta lactamase basically means that these bacteria produce an enzyme that can effectively break down a wide range of antibiotics, including very commonly used antibiotics like penicillin and cephalosporins. And key resistant pathogens of concern in this context are Staphylococcus aureus, Klebsiella pneumonia, and E. coli, as many of you are familiar with. So our research questions, given this prior knowledge, first we wanted to understand, do animal waste management strategies differ based on biodigester ownership? Second, are people without biodigesters also at risk of exposure to resistant bacteria? Since we know that a lot of these households own animals, regardless of biodigester ownership, maybe there are other animal waste management strategies that are also causing these exposures to drug-resistant bacteria. Maybe it's not just the biodigesters themselves. And then third, is the effluent from these biodigesters actually safe to handle? There are a lot of different factors that impact the ability of these digesters to actually inactivate pathogens, including how long the manure is left in there, the temperature, humidity, the number of inputs, and diversity of inputs. So it really varies in these small scale systems. And then finally, we wanted to understand if certain waste management practices and strategies were specifically associated with an increased risk of resistant fecal indicator bacteria along different exposure pathways. So we built on our original qualitative research by conducting a fecal exposure pathway study in Kavre. So we conducted this cross-sectional study across 30 tolls. We originally conducted a census to identify all of the households in the region that might be eligible to participate. And we enrolled 301 households that had at least one child under 5 years old and that owned domestic animals and livestock. And this study was conducted between May and June of 2023. So we wanted to capture a variety of behaviors and infrastructure and strategies around animal feces exposures. And so we kind of broke these up into 4 categories based on data from interviews and observations. The first is animal ownership, then animal confinement, animal waste management, and animal care and contact. Then we analyzed drug-resistant bacteria along key exposure routes. So we collected 3 to 4 samples per household. We had child and mother paired handprint samples, as well as soil from child play areas. And finally, we collected the biodigester effluent, which was sediment that was at the base of the pipe where effluent would have come out. We also quantified fecal indicator bacteria from these samples using IDEX. So for each sample, we had 2 IDEX trays. The first was to estimate total coliform and E. coli overall. And the second tray, we actually spiked with cefotaxime, which is a 3rd-generation cephalosporin. And usually it's an indicator of potential ESBL multi-drug resistance. And so we spiked with cefotaxime and counted the total coliform and E. coli that grew in the presence of cefotaxime and therefore is likely resistant. Finally, just for the soil and biodigester effluent samples, we also plated these samples on selective media to see if we could grow ESBL producing E. coli. And so we did antibiotic susceptibility testing and confirmed whether ESBL grew from these samples, and we enumerated those quantities of colonies. So just a brief context of the households that we enrolled. 78% had improved drinking water, 74% had an improved water storage method, and 95% treated their drinking water. While only 47% had a latrine and hand washing station. And I do want to mention that households that had a latrine were pretty much all of the households that also had a biodigester. So these interventions were sort of a package by the government of Nepal and foreign aid agencies. And so the program would subsidize the construction of both a latrine and a biodigester together. So those were co-occurring for the most part. So 50% of these households had biodigesters, and as I mentioned, these were more likely to have a latrine and hand washing station. There's no difference in household wealth between those who had biodigesters and did not have biodigesters at the time of the study. And only half of the households that had biodigesters had added animal waste to those in the last 7 days. So some of these digesters were not as frequently used. And 18% reported giving animals antibiotics in the last 3 months. So for our first question, do animal waste management strategies differ based on biodigester ownership? We found overall that more cow and buffalo ownership occurred among biodigester households. So in this bar chart here, the light blue bars are households without biodigesters, and the purple bars are households with biodigesters, and we have the percent of households here on the x-axis. And so we can see that households with biodigesters had more cows and more buffalo, and those were typically the animals whose waste would be placed into the biodigesters. It's easier to collect than, for example, chicken or goat feces. We also found that in terms of animal care and contact, households with biodigesters reported more frequently cleaning their housing structures for their animals, specifically for cows and buffalo and for goats. And this is, again, probably just because as they're cleaning, they're removing that animal waste to put into the digester. Interestingly, I do want to mention down here, the chicken feces was cleaned daily more frequently among households without biodigesters. So we noticed that households with chickens and biodigesters were not specifically using their chicken feces in the biodigesters. So they were less likely to be cleaning that feces because they were busy collecting other animal waste to put into the digester. In terms of animal confinement, we identified that most households have separate structures for their animals. So over 75% for almost all species, except dogs, maybe. And especially for chickens, everyone had separate confinement structures for their chickens. However, even though there were structures for these animals, they were still reported as free roaming for a significant amount of the time. So chickens and dogs specifically were left to roam despite having a separate structure for them. Cows and buffalo and goats were more commonly kept outside of the compound. And a compound is sort of just a household and a courtyard and maybe an animal confinement structure. And so that's just where the family would spend most of the time. And outside of these compounds is where cows and buffalo and goats were typically kept. And then chickens and dogs were commonly let into the house. For animal waste management strategies, composting is the primary waste management strategy. And there was, excuse me, about 25% of households did report cleaning feces with bare hands. And a lot of the chicken feces was composted, but it was also placed in fields directly without composting, whereas these other waste types were composted first. and then typically either placed directly in the field or placed into the biodigester after composting. And the compost piles also included other organic waste from cooking and other sources. So the next question is specifically in regards to whether people without biodigesters are also at risk of exposure to resistant bacteria. And so we collected over 1,000 samples at these households. And first, just to orient you here, we have child hand rinse samples in the dark purple, mother hands in blue, and household soil samples in green. And we have the percent positive for total coliform among households without biodigesters on the left and with biodigesters on the right. So here we see no difference in total coliform between the two household types. When we look at drug-resistant coliform, we're seeing much lower prevalences, but again, not much difference between households with versus without biodigesters. The highest prevalence of resistance is in the soil samples, which makes sense. That's probably where most of the fecal contamination is. We were a little bit surprised to see 20% of caregiver hands positive for resistant total coliform. And for E. coli, we did actually find a significant difference between total E. coli on child hands. It was lower among children from households without biodigesters compared to children in households with biodigesters. And we're seeing similar trends here in terms of the contamination being lowest on child hands and highest in soil. And for resistant E. coli, we found lower prevalences again, but no significant differences between biodigester versus non-biodigester households. And finally, looking at the effluent samples, I gave you a little hint of this, but is effluent from biodigesters safe to handle? It looks like there was a lot of culturable E. coli, total coliform, and resistant E. coli and total coliform. So almost all biodigester samples had fecal indicator bacteria, and 72% had resistant E. coli, cefotaxime-resistant E. coli. So because of these results, we definitely we wanted to investigate this further in terms of the effluent samples. Sorry, I think somebody might have annotated on this slide. I don't know if we can remove that. Let's see. There we go. Okay, sorry about that. Okay, great. So moving forward, maybe, there we go. Looking at the concentrations of fecal indicator bacteria and resistant indicator bacteria by sample type. So again, we're looking at the same sample types across the board, but now we're looking at the log 10 MPN, or most probable number of each fecal indicator bacteria. And we're seeing the highest concentrations in effluent samples, but we're still seeing relatively high concentrations across these other samples for total coliform. And for resistant total coliform, we're just seeing a really, really high concentrations among biodigester effluent samples and much, much lower concentrations of resistant total coliform across the hand rinse and soil samples. And similarly for E. coli, we did find similar patterns where biodigester effluent was highly contaminated with high concentrations, both of total E. coli and resistant E. coli. And then with the IDEX data that we had, we were able to estimate the proportion of fecal indicator bacteria that was resistant to cefotaxime. So what we did was we took the most probable number of resistant E. coli and total coliform and divided it by the total number, the total MPN for each of these indicator bacteria to estimate this proportion. And again, we're seeing similar patterns where we see really high proportions of resistant E. coli in biodigester effluent samples that had resistance. We're also seeing some samples with really high proportions of resistance for child hand-rin samples and caregiver hand-rin samples and soil samples. So there were some few samples where there was really high prevalence of resistance um, and yeah, so we'll talk about the potential risk factors for that next. And so we also cultured ESBL producing E. coli, as I mentioned, and we found 20% of household soil samples had ESBL E. coli. And this was not different between biodigester and non-biodigester households. All households had about the same. The prevalence was consistent between the two groups. We then detected ESBL E. coli in effluent samples at a prevalence of about 35%. And this is consistent with ESBL prevalence from household sewage and other recent studies. And this was, again, culturable bacteria. So these are viable organisms that can potentially cause illness and specifically a multi-drug resistant infection. So it's definitely clinically relevant in this setting. So next, we investigated whether certain waste management strategies were associated with increased resistant fecal indicator bacteria along these different exposure pathways. So we used Poisson regressions with generalized estimating equations for cluster robust standard errors. We adjusted for household wealth, which was defined as an asset index, and female caregiver education level and toll, and the reference level for all of the exposures was none or never. And so just to walk you through this forest plot here, we have our odds ratios on the bottom here on the x-axis, and we have our risk factors on the y-axis here. And so at the vertical line, that's a null association. So an increased risk is to the right of that line and a reduced risk is to the left. And again, we have our different sample types color-coded. And so for resistant total coliform, we found that owning chickens was the only statistically significant risk factor for having more resistant total coliform or resistant E. coli in household soil, which makes sense since the chickens were free roaming. Their feces wasn't typically collected as often as other animal types. We also saw that similar pattern with goats and dogs for resistant E. coli, again, which these animals were more likely to be free roaming and their waste was not typically picked up and put into the digester. Interestingly, we did see that cows, there's actually a reduced risk of contamination of resistant total coliform on child hands among households that owned cows. So I'll talk a little bit more about that in a minute, but that was an interesting and sort of unexpected finding. For animal care and contact, we found that handling dog feces with bare hands was associated with a very strong increased, there was a large increase in the risk of contamination of resistant total coliform on child or on mother hands and on household soil. And there are some missing relationships here or some missing effects because the models could not converge due to lack of data in some of these categories. We also found that handling cow and buffalo feces with bare hands was associated with an increased presence of resistant E. coli on child hands. And we also found that child contact with stray dogs was a risk factor for resistance on child hands. And then again, for increased risk of resistance on child hands, we found that when caregivers mixed biodigester slurry by hand, then we found this increased risk of resistance on child hands. And that was a little bit more consistent with our original hypothesis that caregivers were mixing slurry by hand and then interacting with their children, which might increase the risk of exposure to resistant bacteria. We also found for animal confinement that keeping animals in the house interestingly and unexpectedly, was associated with a reduced risk of contamination on child hands. Um, and then when animals were kept free roaming, we found an increased risk of resistant E. coli in household soil, which again was consistent with our hypotheses. And then we found that animals that were always kept outside of the compound, those households had a reduced risk of contamination in household soil. So a lot of these were very consistent with what we expected, but then we found some, like we really didn't expect to see a decreased risk of child hand contamination when animals were kept in the house frequently. So we weren't really sure what was going on there, to be honest. We can sort of, yeah, think creatively about that one. And then finally, for animal waste management, we looked at a lot of different factors, and we have even more factors that we could look at. So we sort of picked the factors that we thought could be most easily modified by different interventions or behavioral changes. So first, we found that households that placed animal feces directly into the field had an increased risk of resistant E. coli in household soil. This is potentially because chicken feces was primarily placed into the field, and households that own chickens, as we know, had more soil contamination. Households that placed animal feces into a compost pile actually had lower prevalence of resistant total coliform on child hands. So perhaps collecting animal waste and sequestering it into a separate area of the compost pile was protective against contamination of child hands if the child was crawling around in the dirt elsewhere, but not near the compost pile. Cleaning dog feces daily was associated with an increased risk of contamination of resistant fecal indicator bacteria in household soil and on caregiver hands. This is a daily or a great reminder to everybody who has dogs. Please wash your hands after you take your dogs on a walk. I was thinking about this a lot last night as I was walking my dogs with my baby. Um, and then finally, we did see that households that added biodigester feces into their or added feces into their biodigester in the last 7 days had a reduced risk of contamination of resistant E. coli in household soil. And so it's very possible that by adding feces into the digester, you are automatically cleaning the rest of your household environment. You're picking up feces from the soil or from these animal confinement areas. So you're reducing any fecal contamination in the soil, but potentially creating there are additional exposure pathways through contact with um, between caregiver hands and feces, and then caregiver hands and child hands. But we did see that there was potentially a decreased risk of contamination associated with using these biodigesters, which was also not exactly what we predicted. So overall, the summary of the initial results was that cow and buffalo waste was typically composted and put into a biodigester. And cows and buffaloes and goats were typically confined outside of the household, and there was more frequent cleaning of these animal structures. And so there were a lot of different waste management practices observed, mostly based on animal species that was owned, and the animal species that were owned was associated with biodigester ownership. So there was a potential link there. We did not find that there was a significant difference between resistant fecal indicator bacteria on child hands, mother hands, or in soil when comparing households with versus without biodigesters. So there were potential risks of exposure in both contexts. So it really has more to do with the handling of animal waste and animal waste management practices rather than just having these biodigesters. In terms of factors that were associated with resistant fecal indicator bacteria, we found that on hands, handling dog or cow and buffalo feces, mixing biodigester slurry by hand, were associated with child hand contamination. And in soil, we found that chickens, free roaming animals, and cleaning dog feces more frequently. These were all associated with more contamination. And then finally, in terms of the biodigester effluent, we were able to culture both cefotaxime-resistant and ESBL producing E. coli in 35% of the samples and 80% having cefotaxime-resistant fecal indicator bacteria. So based on this, I think the biodigester effluent is potentially not safe to be handling because these are viable organisms that can cause multi-drug resistant infections. We don't know if these are pathogenic organisms or not. And so we have some follow-up work that we're hoping to do that I'll talk about in just a minute. So our additional analyses and community-driven solutions that are being generated from this work, first, we were really interested in seeing if there's any effect measure modification by hand washing or community level animal density. So in this area, the households are often pretty spread out, but there are a lot of sort of smaller villages where households are close to one another, and households often utilize or share resources in neighboring households. Sometimes they'll even share biodigester effluent or share, they'll share animal confinement spaces. So there are a lot of community level and village level factors that should be looked at, because even if, for example, you are not necessarily keeping your animals directly in your courtyard or your compound in your household, you might have neighbors' animals that are free roaming coming through. So there are a lot of community level factors that we can look at. And because we collected a census that asked about animal ownership in this area, we do have a good idea of the community level animal ownership in this region. So we can look at that and do a bit more of a spatial analysis to understand the community level effects of contamination. And then, as I mentioned, hand washing. There have previously been some hand washing behavioral interventions in this area, and based on our observations and the study results that we found, there is still pretty low prevalence of hand washing with soap in the area. And so just understanding that a little bit more and how we might see a difference in the results given hand washing after certain key events like stirring in animal waste into these biodigesters or applying effluent to crops before cooking, before breastfeeding, all of those sort of key events or key points of contact where you can intervene, and the hand washing might actually impact the contamination levels after interacting with animal waste. Next, which I'm really excited about, is identifying factors driving the high prevalence of resistant bacteria in biodigester effluent, as well as sequencing and using qPCR to detect other zoonotic pathogens and resistance genes in the effluent. So I just wanted to show quickly this figure here on biodigester practices. So the percent of households that added feces in the last 7 days was about 50%, as I mentioned, but almost 75% of households, regardless of having used it in the last 7 days, were still applying the biodigester effluent specifically by hand. Fewer households were using an agitator to mix the slurry, but not as many people as we thought were using their hands to mix the slurry. So I think what the main driving factor is in terms of exposures and potential risk is really using the effluent on crops, not so much mixing the slurry. Yes, we should definitely talk about other better options like using a stick, fixing the agitators, reducing all contact with the animal waste slurry in that digester inlet. We can come up with some creative solutions to improve that. But I don't think that that's necessarily the most important potential exposure route, given how many folks in this region are applying the biodigester effluent by hand, and given the prevalence, the really high prevalence of resistant bacteria in the biodigester effluent itself. And we do also know that the biodigesters are producing gas pretty consistently. So 75% of households had biodigesters that were actively producing gas, which means that the composting process is working for methane production, but not necessarily for pathogen inactivation. So we really want to understand what are the other sort of structural factors that are not enabling these to function as intended and inactivate pathogens. One really key event that happened during the original study where we identified an association between biodigester use and child diarrhea. That study took place in 2015, and that was when there was a very, very large earthquake in the region. And a lot of the households reported that their biodigesters were cracked. The tanks underground were cracked, or the slurry collection cement area that was cracked, or piping was cracked from the latrine. There was so much damage reported from that earthquake, and there was government assistance to fix a lot of those biodigester, the infrastructure. But we think that, that was already 10 years ago. There could have been a lot of, you know, ongoing maintenance issues and infrastructure issues that could still be playing a role in how effective these biodigesters are at actually inactivating pathogens. That could be really important to understand. Like, what do we need to invest in for these biodigester systems to be safe? How do we make them really safe to use? Because they can be really wonderful interventions. But we just have to make sure that we're thinking about all of the possible risks and benefits associated with them to really maximize their benefits. And so the next phase of this work is, like I mentioned, doing some genomic work and molecular work with qPCR. So we're planning right now follow-up work where we're using these effluent samples. We actually previously extracted DNA from about 130 biodigester effluent samples. And so we're working with our partners in Dulakel Hospital Microbiology Laboratory to develop some methods that are feasible in country, given their equipment that they already have. How can we do sort of a multiplexed qPCR panel to detect a wide range of zoonotic pathogens and resistance genes? We know that there's fecal indicator bacteria, which likely means that other pathogens are prevalent, but we don't know which pathogens we should be concerned about. And so we're going to be testing that out hopefully in the coming months. And so hopefully we'll have some more results to present. And I think that will be really informative in terms of how to address these exposures to animal waste, and what types of pathogens we're seeing and how they're related to the different animal inputs into the biodigesters, and as well as human inputs. And Finally, we're really interested in looking at how these different exposure pathways are linked to health impacts. So for this study, we also collected self-reported health data, both for the children and the caregivers and other household members. So understanding how the entire household is impacted by these different animal waste management practices and using the biodigester effluent on their crops. We have a lot of data on crop production and irrigation methods. And so it's a really rich data set. So there's a lot of questions that we can answer and understand how to improve health and maximize the benefits of these interventions. And so we're also hoping to, once we kind of finalize these results and work with our partners in Nepal to develop some communications materials. We're going to work with our communities to develop interventions that mitigate exposures. So as I mentioned, these could be really simple fixes like using a stick to mix the slurry in the inlet of the biodigester rather than hands. Maybe we can come up with a little bit more of a sophisticated tool, but something that's locally available, that's really easy to acquire, that's not likely to break. So just kind of thinking creatively around that. And then in terms of applying the biodigester effluent to crops, making sure that we're understanding how long we should be waiting from putting in animal waste and letting it sit into the biodigester, making sure that there aren't structural issues with the biodigesters, and fixing those when we can. And then potentially trying to incorporate some use of PPE, protective personal equipment like gloves, when applying these the biodigester effluent to crops, and also improving access to safe, not only drinking water, but also water for washing those crops. And we did see that there was mostly improved drinking water and treated drinking water in the area. So as long as we have sufficient access to that water, these households should be able to wash those crops a little bit more thoroughly and hopefully reduce any presence of bacteria on those crops from the biodigester effluent. We did not collect samples from crops for this study, and that is something that we thought about a lot. We were limited in what we could collect. We couldn't look at all possible exposure pathways for this study, but that is something that we're thinking about for the future because we know now that the biodigester effluent is probably a key exposure pathway. We might look into the impacts of using that effluent on crops and if contamination is continuing into the household and onto people's plates, or if there is sufficient, washing practices and everything. So yeah, there's a lot of great data that we have and a lot of things to look forward to in terms of next steps. And this has just been a really wonderful project to work on. We have amazing partners in Nepal. These are just a few folks collecting some samples in the field and doing some lab work. Hanging out with some goats, and it's just been a really great project. And we appreciate everybody who has been involved. These are some of our students and partners in Nepal and from Berkeley.